Hi, in this lab we're going to be looking at the genetic transformation of E. coli cells using a specialized plasmid that will allow the bacterial cells to glow green. And this is known as the pGLO lab because of the name that has been given to the plasmid. So just to get us started here, let's really quickly review what the central dogma of molecular biology is. In bacterial cells, and it's similar, although with some modifications in eukaryotic cells, in bacterial cells, the DNA is stored just in the main part of the bacterial cell, actually in the nucleoid, but the DNA holds the information, and that DNA is transcribed into mRNA. So that information that's stored on the DNA is written onto mRNA. And then the information on the mRNA is then used by a ribosome in order to create a polypeptide. So the information specifically is used in order to determine the sequence of amino acids that make up the polypeptide. And that job is done by the ribosome in the process called translation. Now, cells can control how much of any gene product they need to make. And as you might imagine, it's important for cells to be able to do this. And so cells have developed ways to do this to increase their evolutionary, their ability to survive in a particular environment. So we can, often for bacteria, we often divide the different regulatory steps into transcriptional control, translational control, and post-translational control. And so in this picture here, you can see that that the DNA in red, the information is being written onto RNA, the yellow strip here, and then that RNA, the information is used by the ribosomes to make proteins, and you can see the proteins that come off it in green at the top right. So if a cell exerts transcriptional control over the production of an enzyme or really any protein, then this is a really efficient way to do it because the cell hasn't made any of the protein, any of the mRNA, and it hasn't made the protein yet. And so as a result, if it says, okay, don't make this protein now, then none of the energy that's involved in making mRNA and then subsequently protein has been expended. And so the cell hasn't used any energy in order to do that. On the other end of the spectrum, if a cell is using post-translational control in order to determine how much protein to make, then it goes through the process of making RNA, and then perhaps even making the protein, but not activating it yet. And this can be really inefficient because it has to go through all of that energy, use that energy in order to make mRNA and then make the protein for a protein that may never even be used. So in this case, it would be a relatively inefficient system because of all that wasted energy, especially if if the protein is never going to be used because the cell determines that it's not needed. The other side of this, however, is there's a sort of a time period that the cell might be concerned with. So for example, in transcriptional control, if the cell is exerting transcriptional control, then the DNA is perhaps never transcribed into RNA. And so if the cell all of a sudden needs that gene product, it needs that protein, then the cell has to go through a relatively long period of time in order to make that gene product. It has to transcribe, and translate and then activate any proteins that it needs. And this takes time. It takes time to do, and so the bacterial cell doesn't have immediate access to that protein. So transcriptional control can be essentially slow to affect changes on the protein makeup of the cell. On the other hand, post-translational control can be relatively quick. The RNA has already been made, and in fact, the protein, the polypeptide, has already been manufactured. It's just waiting to be It's just waiting there to be activated. And so, under certain conditions, the bacterial cell can activate that that protein, and it can be ready to go pretty much right away. So, transcriptional control is efficient but slow, and post-translational control is relatively inefficient but fast. And translational control is somewhat intermediate for both of those. Okay, now I understand that you may not have seen yet any details and learned from the lecture about the arabinose operon. So let's just go briefly through it. An operon in bacteria are a group of genes along with their regulatory elements that are all transcribed and usually translated all together. Okay, so in this case here, there are 3 genes, AraB, AraA, and AraD. And these genes are all required by a cell in order to metabolize arabinose. Okay, so arabinose is a particular sugar that may be available to a bacteria in a particular environment. And so all 3 of these genes are in fact needed by all of these proteins, all of these gene products are needed by the cell in order to metabolize arabinose. But if the cell doesn't have arabinose, then it doesn't want to produce those. It doesn't want to spend the effort and the energy in order to make those gene products, because making them is energetically costly, and it doesn't want to spend that if it doesn't have any use for them. So in order to metabolize it, it needs to transcribe and then translate these AraB, A, and D proteins. Now, there are other parts of the DNA that are what we consider to be upstream from those genes. In particular, we have an initiator and a promoter. Let's concentrate on the promoter for now. And basically what the promoter does is it provides a landing site for the RNA polymerase in order for the RNA polymerase to find those genes, to bind to the DNA, and then subsequently start transcription. Okay? In order for it to do that, in order for the RNA polymerase to do that, it needs to have what's known as the AraC dimer present, and also some molecules of the sugar arabinose. Okay, so imagine that a cell is sitting in an environment with no arabinose. Okay, that cell will not be able to produce AraB, AraA, and AraD proteins because it doesn't have the arabinose, which binds to the AraC dimer. Okay, in contrast, in the presence of arabinose, the AraC dimer is produced. In fact, it's usually produced. The AraC protein is produced, and arabinose binds to this AraC protein, and this complex promotes the binding of the polymerase to the promoter, and the polymerase can then transcribe AraB, A, and D, and eventually this translation produces the proteins needed to metabolize arabinose. Okay. Now, as it turns out, it's a little bit more complicated. You'll learn in the lecture about how the AraC dimer can be both exert positive and negative control over this system. But for now, let's just leave it at this and say that in order to get the AraB, A, and D gene products made, both the AraC dimer and, importantly for us today, the arabinose is present. Okay, now just to switch gears briefly for a moment, in bacteria have one or more chromosomes, and this is what we would normally consider to be the bacterial DNA. But they can also, some bacteria can also have plasmids, and plasmids are also DNA, and as such, they can contain genes on them. Now, the plasmids can be shared from one bacteria to another, what we term as horizontally. Okay, so they can be taken in by a bacterial cell instead of being inherited by the cell. So, since the plasmids consist of DNA, the genes on them can be transcribed and translated just like the chromosomal genes can be. Now, we're going to be dealing with a plasmid called pGLO in this lab, and natively, this pGLO plasmid contained a gene for AraC and also contained genes for AraB, A, and D. Now, between these, the area between AraC and then the genes AraB, A, and D is a promoter. Okay, the promoter here is where the RNA polymerase binds. And the RNA polymerase binding there, remember, depends on the presence of arabinose. So when this plasmid is placed into a cell, it allows the cell, which perhaps otherwise couldn't metabolize arabinose, it allows it to metabolize arabinose, okay, because of those gene products. Now, you'll also see here on the left that there's another gene called BLA, or beta-lactamase. And this gene allows, when it's expressed, when it's turned into, transcribed and translated, when the gene is expressed, it allows the bacteria, which has this plasmid, to produce proteins that allow it to survive on ampicillin. Okay. Now, ampicillin is an antibiotic, and the ampicillin would usually kill the bacteria. However, the presence of this gene here allows the transcription and translation of beta-lactamase, which then allows it to survive on ampicillin. Now, that's the state of the native plasmid. This plasmid has been changed a lot, and actually, when I say native, it's not really native, but we can think of it as native. But the plasmid has been changed a lot, but the most important thing is that the AraB, A, and D proteins here, which allow it to metabolize arabinose, have been taken out, have been basically cut out of this plasmid, and in their place, a gene called GFP has been inserted. Okay, now GFP stands for green fluorescent protein, and when the cell produces this green fluorescent protein, it actually fluoresces under UV light, and so we'll be able to see that hopefully see that in the lab. Now, all of the promoters are the same, and so the expression of GFP is going to work in exactly the same way as the AraB, A, and D genes. Namely, in the presence of arabinose, the AraC protein will bind with the arabinose and allow the promotion of transcription that will produce the GFP mRNA. Okay, that mRNA then gets translated into green fluorescent protein itself. And of course, the pGLO plasmid also has the beta-lactamase, so it can also confer resistance to ampicillin. Okay, so in this lab, we're going to be doing something called bacterial transformation. And bacteria can take up foreign DNA in a couple of ways, but we're going to be transforming bacteria by basically just putting putting a plasmid, mixing the plasmid with the bacteria, and we're going to treat the bacteria with a little bit of what's called a heat shock, and this will basically make the bacterial cell wall open up a little bit and have little gaps in it, and that plasmid can then squeeze inside the cell wall, and the bacteria will take it up, and once it's in there, it will be able to be transcribed and translated to produce gene products. Now I want to say here that even though we might be mixing the plasmid with bacterial cells, most cells that are in our tubes are going to be unchanged. They will not be transformed. Okay, this bacterial transformation is a very, very inefficient process. Fortunately, it's efficient enough that we can get a number of cells that actually do get transformed. And so most cells will be unchanged, but a small number will accept the plasmid, and they'll be transformed, and they'll be able to grow on ampicillin, and they'll be able to express green fluorescent protein, GFP. Okay, so I'd like you to make some predictions about which and how much bacteria are going to be able to grow on different types of agar. Now, very importantly, we can make our agar. We do make our agar with a source of carbon. For the bacteria. So bacteria will generally be able to grow on agar. It's a great food source for at least these types of bacteria that we're dealing with. And we can choose whether we want to put a different source of carbon in the form of arabinose into the agar or not. And we can also choose whether we put ampicillin into the agar or not. Okay, now remember that ampicillin is an antibiotic, and it will kill all cells that do not contain the plasmid, the pGLO plasmid. Okay, so let's look at this first one. This first petri dish does not have it does not have arabinose, it does not have ampicillin, and we're putting bacteria on there that do not have the plasmid. Okay, so I think that you can see here that that because there's no ampicillin, all of the bacteria that we spread on here should be able to grow. Okay, and this will be a massive number. We'll actually see what we call, or we might expect, we'll see what we might expect to see, and we call that a lawn of bacteria because it's very dense. There's just bacteria growing everywhere. In the second petri dish here, we will have ampicillin, but the plasmid, the bacteria, will not have been transformed with the plasmid. Okay, so here we should see that the bacteria will not grow. Okay, because the ampicillin will kill all of it. In the third petri dish, we should see that we'll have the plasmid inside some of the bacterial cells, and so those particular cells will be able to grow even though there is ampicillin. However, we won't get the lawn of bacteria growing because most of the cells will be killed by the ampicillin. It's only a few that will actually take up the plasmid. In the last petri dish here, we have ampicillin and arabinose, and we're putting on bacteria that have been transformed with the plasmid. Okay? Assuming that the transformation is successful and those bacteria have taken up the plasmid, those few bacteria will be able to grow despite the ampicillin, because of that beta-lactamase gene on the plasmid. But importantly, in this fourth one, we also have arabinose. Okay, so remember that arabinose is needed in order to express that GFP. Here in this last one, we have arabinose, and so the bacteria will be able to express that GFP. And so if we shine a UV light on these bacteria, we'll actually see them glowing green. Okay? So hopefully we'll see these in lab and get the predictions that we just set. What I suggest you do is to think about this table and write it down into your lab manual with the predictions that we just made.